Bismillahirrahmanirrahim. Assalamu alaikum class. So our next practical number third and fourth is the floral biology and the crossing techniques of wheat. The scientific name of the wheat is the Triticum aestium crop, and so this is the vegetative stage of the wheat crop. You easily observe the wheat field crop before flowering. This is the wheat variety WH711 at flowering stage, and Allness variety C306. Then the compact air type of the wheat. Compact air type of the wheat is in which the florets are compactly arranged. In this spike we have observed that the florets are Lux air type of wheat in which the florets are distantly present. So in this we have seen that a complete spike of wheat from left side. Then in middle you have observed the lower portion of spikelet, and on the right corner you have observed the top of the floret of wheat or spike of wheat. In wheat spike there are spikelets are present as you observed in middle picture. There are also awns are present, and this is the rachis on which these spikelets are attached. There is extruded anther is observed, while when you in the right corner you have observed that the central floret, then secondary floret and primary florets are also present. There are the lower one, the glumes are present. In the glumes, three florets are present basically. This is the open spikelet of wheat in which the lower side the two glumes are present, in which there are the primary, secondary and tertiary floret are present. So when we focus on this spikelet, there is the open primary floret in which you have seen the ovary, lemma, pallia and anthers. So if we talk about the floral biology, you have observed each spikelet have glumes layer in which three florets are present, while each floret have the two layers. The lower one is lemma while the upper one is the pallia. After pallia the central floret is present. Again the secondary floret is present. Here you observe the bifeathered feathery stigma is present. This is a reproductive organ, anther and stigma. This is the close-up view of reproductive organ. You have seen the anther, the stigma, the ovary and the filament through which anther is attached. This is the close-up view of gynoecium or the female part, in which you have bifeathered stigma and ovary is observed. Now this is the structure of one floret in which the lemma, pallia are present, lodicules through which this floret is attached, and awns are present. So this is the basal sterils. In each wheat spikelets there is the lower side of the spikelets are sterile, and also the terminal spikelets of the wheat showing the extruded anthers. This is the basically emasculation wheat stage on which these anthers are yellowing in color but surround the ovary. This is the parts of spikelet of floret after fertilization. When the fertilization takes place, then this is the start of grain formation. This is the developing grain in the ovary. This is the grain structures. In this floret number 2, the stigma, anther and spikelets of wheat are easily observed. So how the anthesis take place, and what is the anthesis? Anthesis is basically the maturation of anther when the pollen grains start forming inside of anther. In wheat, the anthesis start from the upper two-third part of anthesis started from the two-third part of the wheat and protrude towards upper and lower side. In this picture you have observed This is the floral biology, and now we will move towards the emasculation, pollination, or crossing techniques. In this case, emasculation, the open glumes and remove the central floret and cut. In emasculation, you have firstly remove the glumes and remove the central spikelets like this. First of all, we cut this glume, right side glume, then left side glume, then we remove the central floret. Now we have moved the remaining two floret on the upper 1/3 portion of two floret. This will seem like this. Then anthers will be removed with the help of forceps. Then we bag it. This is the actual process of emasculation pollination in field. This is a crossing block. So, what stage is the correct for emasculation? This stage is correct when the half spikes are inside of the flag leaf. Here we are removing the central floret with the help of forceps. Then the anthers are removed with the help of forceps. The ovary will be seen. So, this is the emasculated spike. We will cover the spike after emasculation due to the contamination of foreign pollens. When we how we prepare the male parent, the male parent will be taken from the plant and cut it with the help of scissor, then protrude out in the soil, then we stuck in the soil at sun bath with the help of sun or the sun energy. The anthers protrude out from the floret, and the anthers begin as like this. Sun exposed, the anther protrude out, anther exerted out of the floret. This is the pollen grains, and this is formation start. Close up view. So, how to pollinate? The pollen bearing spike inverted on the emasculated plant and bag it like this. We have drawn the date of emasculation pollination on this. This is the crossing block of CMS lines. That's all.